Bismillahirrahmanirrahim. Today we are going to talk about the largest internal organ called liver next to your skin. It is present in your right upper quadrant of the abdomen below diaphragm. It's being covered on its outer side by a thin connective tissue capsule made up of dense irregular fibers called as glycan capsule. This capsule is sending septa inside to divide your liver into lobes and lobules. This capsule becomes thicker near the hilum where entry-exit of vessels, lymphatics and ducts are there. Now we have to see that there is a left lobe and the right lobe, which are the major one. In addition to them, it has got two smaller lobes, posteriorly and inferiorly, called them as quadrate and quadrate lobes, right? This liver, which is almost 1.5 kilogram, or you can say 2% of your body weight, receives dual blood supply, right? What I mean to say that it receives its blood supply through the hepatic portal vein and the hepatic artery. The portal vein is going to supply almost 70 to 80% of the blood, and this all blood comes from stomach, intestine and spleen, so definitely deoxygenated. And the blood that is coming through hepatic artery is 20 to 30% and is well oxygenated. So your liver is supposed to absorb materials via intestine that has to reach liver through your portal vein, except the complex lipids like chylomicron that are being transported via the lymphatic vessel. Your liver has to do a lot many functions, and the special beauty about it that its single cell does the two function bilaterally, in a sense that it is exocrinal in nature and endocrinal as well. Let's talk about the endocrinal and exocrinal function of your liver briefly. So as an exocrinal function, it has to produce bile, which is needed for digestion and GI antimicrobial action, right? And in addition to it, you can see that it has to perform its various other functions, like it has to maintain your blood sugar level by the storage of glycogen, right? Then it has to store various micronutrients, you can see in the list, the minerals copper, zinc, magnesium and iron. Vitamins include A, D, E, K and B12. The list is of the fat soluble, and B12 is the water soluble vitamin, right? Now we have to see that it has to involve itself in the production of the cholesterol, which is a precursor to your various sex hormones, and vitamin D. And what else? The major function of your liver is protein synthesis, right? Like by producing prothrombin, it takes part in blood clotting, and lipoproteins are supposed to be a part of your cholesterol transport. Then immune function definitely through globulins, and your oncotic pressure is maintained by the albumin. Then copper bioavailability is via ceruloplasmin. Hepatocytes deal with the detoxification of drugs, alcohol, fatty acids, steroid hormones, ammonia and various environmental toxins or allergens. Metabolism includes the conversion of thyroid hormone T4 to T3. Then detoxification of fat is also there. And in immune system, it has to deal with the viruses and pathogen in maintaining the hepatic and portal venous immune system, right? Now let's talk about the cell of the liver called hepatocyte and its organelles. You can see its basal surface, you would say, and you can watch the nucleus present near the base having the nucleoli in it, right? And on its apical surface, you can observe the microvilli. And now we have to focus on various organelles and their function as well. Now visualize that the nucleus is having on its margins nearby the abundantly present rough endoplasmic reticulum stud with the ribosomes. What they both have to do? They have to synthesize various plasma proteins, and in addition to it, they give basophilic stain to cytoplasm as well. Now you can visualize that there is the smooth endoplasmic reticulum abundantly and evenly distributed throughout the cytoplasm of the hepatocyte. It has to deal with the enzyme system for detoxification, you would say, and biotransformation of various substances, like it does conjugation of the hydrophobic bilirubin to water soluble, you would say, bilirubin glucuronide, and being excreted into bile canaliculi. And this also is dealing with your oxidation, methylation, and conjugation of steroids, barbiturates, antihistamines, and anticonvulsants, right? And some other drugs. Important and interesting fact about it, that if the drugs, they are staying in your body prolonged period, for a prolonged time, right, what happens, that there would be the increased amount of smooth endoplasmic reticulum in hepatocyte, leading to improved liver's detoxification capacity, right? Now, if the glycogen granules and lipid droplets are present in hepatocytes, and you see the ferritin complexes, they are meant to store temporarily the glucose, triglycerides, and you would say iron respectively. And if you can concentrate on the peroxisomes, which are also abundantly present, they do oxidation of various, you would say, fatty acids, leading to the breakdown of hydrogen peroxide. In this way, they convert excess purines to uric acid. These functions and related organelles are important. That's why we are mentioning them. Then you can watch the placement of the Golgi complex or Golgi apparatus, supranuclear position, right, involved in the synthesis of both plasma proteins and bile component, a very important function of the liver. Then you can watch the purple color structure. These are numerous mitochondria. They are meant to provide energy for all the activity of the hepatocytes, right? Now we are watching the classical liver lobule, which is hexagonal prism shaped structure, right, and being traversed in its center by a structure called as central vein, which is a tributary of the hepatic vein, right? And you can visualize that there are the plates of hepatocytes being radiating like sun rays from center towards periphery, and they are making one cell thickness, right, and they are separated from each other by the hepatic sinusoids. These blue color structure, if you can visualize, radiating in the similar way, radially arranged around the central vein, and they are called as hepatic sinusoids, right? And now at the angle of that hexagonal classical liver lobule, there are triangular areas called as portal spaces, portal areas, or why they are pressed here. They contain three tubular structure, as you can see in the picture, including a branch of a portal vein and a branch of a portal artery, and the third thing is the bile duct, and collectively they are forming the portal triad, right? An important fact about the liver classical lobule is that the blood flows from the periphery towards the center, right? The direction of blood flow is from periphery towards the center, right? And another interesting thing is that the bile, which is being produced by the hepatocyte, and the flow of bile is from the center to the periphery. The hepatocytes are producing the bile, and they have to pass through the minute channels, the bile canaliculi, and the direction of flow of bile is from the center to periphery, means just opposite to the flow of the blood, right? These bile canaliculi are nothing but a minute narrow channels, you would say, marked in green color, right? And they are present or located between adjacent hepatocytes, right? In the liver cords or plates, you would say. And they are simply spaces, I would say, present in between these hepatocytes and have no separate epithelial lining, rather placed in between the adjacent hepatocytes, right? As we mentioned earlier that there are three ways to study the liver parenchyma. First, we studied the classical liver lobule. Here I can mark it again for you. That's the classical liver lobule. That is meant for studying the endocrinal function, right? Then there is another way of studying, that is the portal lobule. That portal lobule is meant to study the exocrinal function. Then we can see there is another structure which is called as hepatic acinus, and it has to deal with the you would say to study the nature of the blood supply to the liver. So these are the three ways to study. Now come towards the portal lobule. If you focus, it's a triangular area to study the exocrinal function, and we have to focus on the production of bile here, right? And what we can see here is that in its very center, this is the portal area and consists of the liver tissue that drains the bile into the bile duct of the portal area. Now we can watch that it's a triangular area in cross section and contains three parts having central veins on its corner, and in its center it has got the portal area, right? And there is the third way to study the functional unit of the liver is the hepatic acinus. Here you can see a diamond shaped cross sectional view in both these picture. I would suggest this is the diamond shape, this is another diamond shape. These are both hepatic acinus. Here what is gonna happen that one central vein is located on its two opposite ends, or say corners of your diamond shaped area, and in its very center you can see that this central area, if I can mark it for you, that is having a branch of portal vein, hepatic artery, and a bile ductule, means it is a portal triad or portal area in the center, and on the corner of the hepatic acinus two central veins are located, right? This is another picture to show you the darker brownish color are the hepatic cords made up of hepatocytes, and in between the yellow areas are the hepatic sinusoids, and this hexagonal shape is addressing on its six corners rather the portal areas, carrying the three structures forming portal triad, right? Now we have to talk about the hepatocytes, which are again polygonal in shape. Let me mark an individual one. They are closely packed as an anastomosing plates of cells arranged radially around the central vein. You can see the central vein here as well, right? And in between them there are hepatic sinusoids, right? And what else you can see, that these hepatocytes are having a single spherical nucleus, but sometimes in 20% of them they may carry two nuclei, and they vary in size than ploidy, and usually they are having large nuclei which are polyploid, sometimes tetraploid, and larger nuclei sometimes having 64 n amount of DNA, right? Here you can see the ultrastructure of the hepatocyte being drawn schematically, right? And the nucleus we discussed having the sometimes two prominent nuclei. The special conspicuous feature of hepatocyte nuclei is the presence of scattered clumps of heterochromatin, right? And under electron microscope, you can see various organelles we mentioned earlier. Let me enlist again. They could be having mitochondria scattered throughout. So many small Golgi complexes could be there. Microbodies like peroxisomes and lysosomes are abundantly present, right? And there is abundant mitochondria that is imparting acidophilic stain along with that of the smooth endoplasmic reticulum. And the basophilic stain, you must be knowing, is imparting by the rough endoplasmic reticulum along with the ribosomes. And you expect so many glycogen and fat droplets inside that hepatocyte. So let's talk about the surfaces of hepatocytes. We consider that it has got the lateral side or lateral domain and the sinusoidal domain. What are these? Sinusoidal domain is that surface which is facing towards sinusoids, right? And they are showing the microvilli in them to increase the surface area. You can see, right? Projecting towards a space called as space of Daisy. That space of Daisy is present between the hepatic sinusoid and the row of the hepatocytes, right? And you can visualize the lateral domain which are present among the adjacent hepatocyte, right? And we can observe the greenish color structure, the bile canaliculi being minute one without having their own epithelium, right? To explain the space of Daisy, I have to consider the classical liver lobule again. You can see the central vein and the pinkish color hepatocytes radiating like a sun rays, right? And you can visualize the central marked the sinusoids. These are the sinusoids, right? And these are the hepatocytes. And where is the space of Daisy? This space of Daisy, let me mark again, is present in between the plates of hepatocytes and the sinusoids. Means a space present in between the bricks of hepatocytes and the blood channel, the sinusoids. These are the space of Daisy, or called as perisinusoidal space, the self-explanatory term. What's they carry? They usually carry star-shaped cells called as hepatic stellate cells. They are also called as itocells, and they are supposed to store the vitamin A. They are being mesenchymal or in origin, and they basically are supposed to store lipid droplets and also called as fat storing cells, right? And usually if a pathological condition occurs like cirrhosis, the normal collagen protein type 3 changes, and the cells now gonna be there, but the collagen production is now protein type 1 and 2 and being deposited in this space of Daisy and leading to the fibrosis of the liver, right? Now we have to talk about the duct system of the liver, which we discussed in the gallbladder as well. You can see the brownish color hepatocytes, and in between the minute greenish color structure, the bile canaliculi, the smallest bile channels, right? They are being formed by the plasma membrane of the adjacent hepatocytes. So they don't have any separate epithelium. They are joining to form a little bit larger canals called as bile canal of Herring or bile ductule sometimes, and they again merge to form interlobular bile ducts, right? Means bile canaliculi are there, then canal of Herring are there, then bile ductules are there, then they are supposed to form bile ducts, and finally they are merging to form right and left hepatic ducts, And if you can recall, these two right and left hepatic duct, they were supposed to form the common hepatic duct. And another important thing is that this canal of Hering is being lined with the cuboidal epithelium, you can see, and they behave as a hepatic stem cells because they have the capacity to proliferate and differentiate into the hepatocytes, right? Now we have to talk about the hepatic sinusoids that are present as a spaces between the plates of hepatocytes, right? And they are lined with the incomplete basal lamina or are fenestrated capillary-like structure, and they are having two types of cell in them, being endothelial cells they have and the Kupffer cells. You can find them, these cells, with the branching processes. The epithelial endothelial cells, you would say, show fenestra or pores in them, right? The blue color marking with the endothelium, right? And they are not closed by diaphragms. Intercellular gaps are there, and you can find the Kupffer cells as well. These Kupffer cells are also called as sinusoidal. Let me mark them first. These brown color, these are called as sinusoidal macrophages or large phagocytic cells, right? And they belong to mononuclear phagocytic system. They are stellate shaped and interspersed, presently you would say present among the endothelial cells, and a permanent resident of your liver cells. Their processes are extending into the lumen of, you would say, the hepatocytes, right? And what they have to do, these Kupffer cells, they engulf and break down the effeted or aged erythrocytes, and this function greatly enhanced after splenectomy, right? Means surgical removal of the spleen leads to their important function which becomes enhanced. They do phagocytosis and eat up the bacteria that usually have a chance to enter the portal circulation through large intestine, right? Now we have to talk about the blood supply of the liver and a little bit about the lymphatic vessels. So for that, first concentrate the space of Moll or the periportal space. That is a small space present between the peripheral hepatocytes and the portal triad area that carries lymphatic vessels. Now after that, we have to talk about the blood supply, which is through the portal vein and hepatic artery, being 70 and 30% respectively. And the branches of that portal vein, they are supposed to give rise to the large interlobar branches that further give rise to the interlobular branches, right? They have to run in the portal canal, and then they give rise to distributing veins, right? And from the distributing veins, they give rise to small terminal inlet venules. Then they directly drain into the, you would say, hepatic sinusoids, and then the blood flows in the central vein, right? This is the central vein, and from the central vein the blood goes into that horizontal lying sublobular vein, and you would say the sublobular veins, they converge to form two or more hepatic veins, and finally their fate is that blood is supposed to go into the inferior vena cava, right? In the 30% blood that has to reach the liver through the hepatic artery, well oxygenated, you would say, it will be giving rise to interlobar arteries that run in the portal canal, right? That interlobar arteries will give rise to two sets of arteriole, they would say, right? And what happens? One set directly opens into the hepatic sinusoids, as you can see, and the other set break up into capillaries and also supply the interlobular connective tissue of the portal canal, and finally drain into the interlobular branches of the portal vein. So that was about the supply of blood that is from the periphery towards the center, right? Like this. Now we have to learn about the hepatic or liver acinus as a structure unit to study the blood supply in the various areas. So kindly concentrate. You can see that there is a zone 1 marked with the number 1, which is at the peripheral located hepatocytes. Then you can see the green color marking as the zone 2 and showing the intermediately placed hepatocytes, right? Then you can concentrate the zone 3 hepatocytes, which are farthest from the, you would say, peripheral blood and is closest to the central vein. And as we know that the blood is going to irrigate these all lobules from the periphery towards the center. So what important about the zonation? Concentrate first to the zone 1. This zone 1, which is closest to the blood supply as coming from the periphery, right? And the zonation will tell us that this zone 1 hepatocytes will receive oxygen first, nutrients first, and definitely toxins first, as blood is coming from the sinusoidal blood, and are first to show any morphological changes if there is bile duct occlusion or stasis. And the cells of this zone will last to die. Why? Because circulation here is present almost all the time, and hepatocytes present here are first to regenerate, right? We have to learn the importance of these zone 1 hepatocytes. On the other hand, if you concentrate the hepatocytes present in this yellow area or zone 3, these cells will show first to observe any ischemic necrotic changes like lobular necrosis because of reduced perfusion here, and they will be showing first to show any fatty accumulation over there, and these cells will last to respond to toxic substances and bile stasis. So we will learn from the division on location of the hepatocytes that which cells receive what first and will be engaged in which functions, right? And cells of the zone 2, their function and morphological characteristics definitely show intermediate to those of the zone 1 and zone 2. So these zonation basically important to describe and interpret the pattern of degeneration, regeneration, and the toxic special effects on the liver parenchyma in relation to the degree or quality of vascular perfusion of these hepatocytes, right? Now I'm showing you a slide of a patient with congestive heart failure. What is gonna happen? You can see that is zone 1 near the periphery, that is the intermediate zone, and that is a zone 3 near the central vein, right? So see the changes in the zone 3. What happens? That the liver acinus here is the first to be affected, right? And hepatocytes here are last to receive blood. You know the reason. And here what is gonna happen, that they undergo ischemic necrosis, and we observe here multiple round vacuoles showing the lipid accumulation here. You should know the reason right now. Now I want to show you the real-time histological view. Here you can see the classical liver lobule with the central vein, and the septa is being marked in the mammary as in stain as dark blue. And in human, let me tell you that connective tissue is not well marked. So in other mammals you will find it very clear. These pinkish cord-like structures are the hepatocytes, and the whitish intervening spaces are the sinusoids. And on the margin you can observe the portal triad that I would mark it. This is the portal vein branch that could be the arterial and biliary structure, right? These are some other picture in which you can hardly find any connective tissue between each classical liver lobule, but you can visualize the central vein and the portal triad. Let me tell you, this is one of the slide identified very easily through its classical pattern, but to learn it is a little bit tricky, right? This is another histological picture of the radiating hepatic cells with the intervening sinusoids of white color, and this is a little bit enlarged view in which you can visualize the hepatocyte with its nuclei and nucleoli. Sometimes binucleated hepatocytes could be there, and all the cell organelles, lipid droplets, all the lot is in its cytoplasm, right? Now this is the slide you will observe in your lab, showing the classical liver lobule. The central vein is there, the septa or connective tissue are there, and you can visualize the portal area. So concentrate the single bricks of wall like structure, the hepatic cords, you would say, made up of hepatocytes, and the intervening whitish area are. Now we have to study the microanatomy, physiology, and the pathology of the liver lobule. And for this, there are the three ways of considering the liver lobule. Number one, this hexagonal look, which is classical hepatic lobule. This is dealing with the endocrinal function. When we study the endocrinal function, we consider the classical hepatic lobule because we know that blood flows from the periphery towards the center. Now come towards this portal lobule, which when we are considering the exocrinal function of the liver cells, that is its bile secretory function. Now here what is going to happen, that the portal area is in the very center and the three triangles are touching the central vein of these liver cells, you would say. And now this portal lobule in a triangular shape area you can very well observe, because we know that the flow of the bile is from the center towards the periphery. From this picture we can see the direction of the flow from all the three sides, so we consider this portal lobule area. Now there is another picture you can see in which the oval shape or diamond shape lobule is there. This is called as hepatic acinus, in which on two sides you get the portal areas and on the two corners you will find the central vein. Now for the study purpose, you can see the classical liver lobule is showing this initial zone. This zone is called as zone 1, which I have marked in pink color. Then next to it in the yellow color you can see the zone 2, and then there is a zone 3 which is very closer to the central vein. Now zone 1, zone 2 and zone 3, these are the zonation showing the location of the hepatocytes. The hepatocytes are called as periportal hepatocytes. Definitely they are nearest to the hepatic artery, so here they get more oxygen and nutrition, so can mostly carry out function requiring oxidative metabolism as protein synthesis. That was all about the zone 1. Now the cells of the zone 3, the blue color, you can very well see here what happens. Hepatocytes are near to the central vein, they get least oxygen and nutrition, and these are the sites where the glycolysis, lipid formation and drug biotransformation takes place, and these are the place of those hepatocytes that undergo fatty accumulation and ischemic necrosis. The intervening zone, which is the zone 2, has got intermediate range of metabolic function. So now you can very well see that the hepatocytes basically these cells adapt to the microenvironment produced by the supply of the blood towards them or to which they are exposed. So now far you are very much clear about the three lobules, the classical hepatic lobule and this portal lobule and the hepatic acinus. Thank you.